Thank you for the invitation, and thank you so much for including me into the program. And I will be talking to you about the cellular and molecular basis of upper motor neuron vulnerability. As you know, HSP, PLS are the diseases primarily of the upper motor neurons. And these are the neurons that reside in the brain, and they initiate voluntary movement. They control and modulate voluntary movement. And in patients, these neurons show primary vulnerability, whereas other neurons in the brain are relatively healthy. So we want to understand what makes them upset, and hopefully we will make them happy again. But first we have to understand what makes them upset. Before I start, I always want to thank people in my lab, in case I run out of time and I can't thank them enough. We have a very active group of people, and we are working very hard in the lab, and we have PhDs, MD PhDs, PhD students, and it's a very active and dynamic lab. And of course, we have many collaborators, and without collaborators, we wouldn't be able to achieve what we achieve in science. It's all about connections, like the brains, like the neurons in the brain. And we receive funding from many different foundations. I received funding mainly from Les Turner ALS Foundation and ALS Association, and we have R01 or NIH grants as well. So as the outline, I can tell you that I have a lot to tell, because we've been discovering very important information over the past 5-6 years. And the outline has 7 points, and I hope that I'll be able to go through them one by one. First, I will tell you a little bit about the importance of upper motor neurons, because you will be surprised that even when I talk to neuroscientists or neurologists, that I have to convince them that the upper motor neurons are important. So I think it's important that we emphasize their unique structure and function, and also try to understand why they become vulnerable. There are intrinsic reasons, there are extrinsic reasons. Intrinsic meaning that happens inside them, extrinsic meaning that are happening around them, and so that they are affected. And what is their mode of degeneration? As Dr. Bass was saying, what is the mechanism? And the mechanism may be different from patient to patient. And then I will tell you a little bit about our drug discovery efforts using upper motor neuron survival as a readout. And I can tell you, even today, for diseases in which upper motor neurons degenerate, there is really no preclinical study that asks the question, do the upper motor neurons like this compound? Nothing. So we try to overcome this, and we try to develop a drug discovery platform, and I'll be talking to you about that. Then I will tell you a little bit about our drug biomarker studies, because without a biomarker, you can't get any of the drug companies to be interested in your research, because they want a quantitative, statistically significant analysis that they can measure, and it doesn't vary from patient to patient, something to hold on to, something that is an outcome measure that they can trust. So without a biomarker, it is very hard for us to engage with drug companies, and that's why we are working on a biomarker as well. And I will talk to you about this. The other one is the translation, because if we know the mutation, maybe just we will give the neurons of interest using, for example, AAV-mediated gene delivery approaches, the unmutated gene or the protein that they need based on the mechanism that is affected. And if I have time, then I'm going to talk to you about out-of-the-box approach, which takes genes to proteins to mechanisms to find the common pathways, and that one drug would be actually working for many diseases, not just one disease. Okay, so if I start, and some parts I'll go really fast, because those are published data, and it's very scientific, so I'll go very fast. Some slides I'll be much slower. But this selective motor neuron vulnerability is actually a very important topic in the field, because in our brain we have millions and millions of cells and neurons, but in diseases not all cells or all neurons are affected in the same extent, to the same extent or degree. Some cells or some neurons show initial vulnerability much earlier than others. And for example, the upper motor neurons, we call them the corticospinal motor neurons in the mice, and they are bed cells in patients, but they are basically upper motor neurons. And they are clinically relevant, of course, and they progressively degenerate in PLS and HSP. And we consider them as the spokesperson of the cerebral cortex when it comes to motor function. Because imagine that these neurons, they have a very long if I can show you here, yes, they have a very long apical dendrite here. They have a very long apical dendrite. Here's their cell body. And if this is their cell body, imagine that this axon goes all the way to Florida, because they have one of the longest axons, right? Like, I'm not a very tall person, but the neuron in my brain is right here, and the axon goes all the way to my spinal cord, to the sacral regions. So the size of the soma, it could be 50 micron, but the length of the axon could be 1.5 meter or 1 meter. So then imagine 20 micrometers or 30 micrometers to 1.5 meter. That's thousands and thousands of different, thousands of magnitude difference, right? So, but these neurons also have these apical dendrites, and this is the brain, and that region is very important for their function, because this is exactly the region where all other cortical neurons converge onto them, right? And by converging onto them, they tell corticospinal motor neurons what to do. So they collect all the information from different regions of the brain, like thalamocortical projections, callosal projection neurons, and different regions of the brain. They all talk to these upper motor neurons, and the upper motor neurons integrate all that information, and they say, this is what you want me to tell you to the spinal motor neurons. Okay, I'll carry your message. And they start an action potential very precisely to the spinal motor neuron in a very distinct region in the spinal cord. Because when I speak and when I move my left arm, I'm not moving my legs, I'm not moving my right arm, right? It is very precise movement. And when I do this, it is because the neurons in my cortex, which project to the spinal motor neurons, indeed in the very precise region of the spinal motor neuron, project to the spinal motor neurons, and then spinal motor neurons make that movement. But it is very orchestrated. And that is thanks to their ability to collect, integrate, translate, and transmit that information. So in diseases like HSP, PLS, they lose this ability. So then the connection from the brain to the spinal cord is lost. The spokesperson of the house is silent. So the connection of the brain to the spinal cord is cut. So the brain cannot really have too much input. That's why we can't move as we wish to. How do we study these neurons? Because brain is complex. It's very heterogeneous. There are thousands and millions of other cells, and it's very hard to see them. So in our lab, we developed many ways. One of them is retrograde labeling, transduction, genetic targeting. So we published a review. I'm not going to go into detail, but basically, we tried to put a fluorescent bead into the corticospinal tract so that we can make these neurons fluorescent. Because as Dr. Buzz said, this is the corticospinal tract. It's very long. And these neurons are the only neurons in the brain that connect all the way to the spinal cord. So if you put a bead, these beads can be taken up, making these neurons fluorescent. So this was an old method, and you have to do surgery, so that's very hard. So we actually engineered a mouse model in which upper motor neurons are fluorescent. So that's very useful because now the neurons that degenerate in patients are fluorescent in the mouse. And this is also published. I'm going to go very fast now. So these neurons, they become fluorescent, and you can see that they are fluorescent all the way from the day they are born till very old ages. And they are in layer 5. They are large pyramidal neurons. Their projection field is correct. So you can see this when they are P0 and when they are P30 here, right? And we confirmed that these are indeed the upper motor neurons by retrograde labeling, by molecular marker expression, by electrophysiology. So this is all published. I'm going fast. And at the end of the day, we actually made the first reporter line for upper motor neurons. And that was a big discovery in the day, because when you see it, you can begin to appreciate and understand and observe. So now that we actually can see them among many other cells and neurons, we will use it to understand why upper motor neuron become diseased. As Dr. Bass was saying, the use of model systems are very important. And as you can imagine, we will be crossing them with different disease models, right? So far, for example, for motor neuron diseases, we had the SOD1 mice, we had the TDP mice, we had the Alsin mice, and actually in collaboration with Dr. Bass and Gerardo Morfini, we are also crossing them with the Spastic mice to make the upper motor neurons GFP in those mouse models. So that is very useful, because now this work is showing the SOD1 mice. When you cross them, you will see that the upper motor neurons become fluorescent, and you pay very close attention to them at a cellular level, right? So this allows us to go from mice to neurons. And then we begin to realize why they degenerate. And as the axons degenerate, right, we were thinking about microtubules, the axonal degeneration, the apical dendrites also degenerate. And you can see that the extension that they have towards the top of the brain also gets degenerated. And that's very important because they lose the connection. They cannot be modulated. They become silent. And that is one of the worst things that can happen to a spokesperson. Then they don't have a message to carry. We, in collaboration with our friends, we also looked at the profilin mice, which is also important for actin cytoskeletal dynamics. And we also find that upper motor neurons degenerate in them. And we looked at the alsin knockout mice and crossing them with our reporter line. We showed, this is also published, that neurons can be fluorescent, which is nice. And because these neurons are GFP is green fluorescent protein. Because these neurons are fluorescent, the beauty is that you can actually do an immunocytochemistry for GFP, and then you can do electron microscopy. Electron microscopy is one of the finest, gives you the finest resolution for imaging. It's like nanometers. You can see organelles. You can see atomic structures. Like it is actually very precise imaging. So then we begin to observe diseased neurons and healthy neurons, and how come they are different, even in the same brain, same mice. So we find, together with Marco Martin and Gabriela Shekarkova, that one of the defects was the Golgi apparatus. And Golgi is important for post-translational modifications. They act at sugars, remove sugars. It's like the dress that the proteins put on. But then if you don't do that right, their folding also, they are more prone to misfolding, and they also may not function properly. We also find mitochondrial problems. The mitochondria either falls apart or they combine together. All right, and sometimes they fuse with the lysosome, which means they're going to be degraded soon. So most interestingly, also look at this. This is the bed cell, huge bed cell, and look at all these gaps that they have in their apical dendrites in their soma. Again, they cannot really maintain their cytoarchitecture. Their apical dendrites are degenerating. They have big holes in them. So we think that in the absence of alsin, looking into this, we begin to understand what's going wrong. It is that they can't really have mitochondria, healthy mitochondria, which means they don't have enough energy probably, and that Golgi's problem. So then you know that their post-translational modification is a problem, and their cytoarchitecture is a problem. And that may be one reason that when patients have alsin mutations, they develop the disease very early because they have many problems. Many problems that they face. So similarly, we also looked at with TDP, and I'm going to go pretty fast, but we find that within the context of TDP, there are, again, mitochondria, ER, and nuclear membrane defects. And if you look at here, this is actually pretty much showing that this is the nuclear membrane. Here is an accumulation on the nuclear side, and this is the cytoplasmic side, that they cannot really go. Can you see? And they're stuck. So there's this nucleus to cytoplasm transport defect. And we can see it very precisely in those images. And again, we made the reporter line. We crossed them to the TDP mice. We know that they actually show progressive corticospinal motor neuron loss. And then these mice also have a phenotype. So that's a great model. And then in these mice, the degenerating neurons are GFP. You can see that the degenerating neurons, their numbers change. So they really progressively degenerate. And then because of the tools that we have, we begin to understand why. And as I told you, it's because of the nucleocytoplasmic transport defects. And again, the mitochondria comes up. The mitochondria is fallen apart. They have gaps in them. And look at also the long mitochondria along the apical dendrite. Normally, here's the, let me see. Yeah. Normally, you would have a very small mitochondria right here. Yeah, this is the apical dendrite. and this is the mitochondria in the diseased one. It seems as if like they really need energy, and the mitochondria also cannot really divide properly, and they extend and stretch. So there are energy problems. And most interestingly, these are the brain samples obtained from post-mortem human tissues, and we were privileged to have some of these samples, and we actually saw exactly the same pathology as we have seen in the mouse, because you can see the mitochondria, it's all inside is all hollow, and this is exactly what we observed with the mouse. Then we looked at the ER, and ER seemed to be enlarged. Then we looked at the human. Look at this ER. It's like a balloon. Normally, this is how it looks. And here, this is how it looks in the human patients. So when you have an ER like this, you cannot have a normal protein production. It's obvious that they are having ER stress. It's obvious that there's something wrong with the protein production and protein folding and so forth. So we, as I told you, now we actually begin to understand what are the problems that are occurring in the upper motor neurons. And so thanks to the reporter lines we have and the techniques we have in the lab, we begin to understand why they become vulnerable. And again, the cytoarchitectural integrity, ER stress, mitochondrial dysfunction, and Golgi apparatus defects seem to be one of the causes. And it may be that different causes are actually more prevalent or more profound in different patients. And there are ways actually to find which patient undergoes which problem more, and I'm going to come to that. So I told you that these neurons are fluorescent, right? Because they have fluorescence they have GFP in them. Okay, so there is a technique which is called fluorescence activated cell sorting, FACS. This technique was used in the cancer field, in the biology, in other areas for many, many years, but nobody really used it in the neuroscience, and I was one of the first ones to use it when I was at MGH trying to purify the upper motor neurons. So we developed this technique to purify corticospinal motor neurons from the complex and heterogeneous structure of the cerebral cortex. Now that they are fluorescent, we can actually put the gates where they need to be and purify them alone. And that's wonderful because, as I told you, in the brain there are many, many different neurons, but only upper motor neurons show primary vulnerability. So rather than studying everyone as a group, we pick only the ones that are affected, put them in one corner and say, tell me why are you upset, rather than asking everyone in the group. Do you understand? So that's actually very important for us to get clear answers. And of course, we have done RNA Seek, and this is one example to show in a color-coded way that we can even see splice variations in different cells. And we have also done proteomics, because after you FACS purify them, you can do proteomics to see which proteins they have. And you can imagine this is a massive undertaking because we have different mouse models, right? So we have SOD1, TDP, alsin, profilin, and we have different time points, P15, P30, P60, P90, and all these time points correspond to different stages of the disease. Very early, like nothing, like P15 is when they are still with their mom's eyes are closed. So they are like teenagers, they're like they're children. And then P30 is when they become teenagers, but they still dependent on you. And then P60 is adults, and P90 is more adults, but not too old. So we have all these different time points to see what is the progressive change, what is the dynamic change over time. And again, as I told you, we do RNA Seek proteomics, and I'm going to tell you some of those results. It's unpublished, so keep it as a secret. So we have looked at P15, and we have numerous genes that are upregulated, downregulated, go up, go down, and we look at the cellular functions that are affected, and with the p-values. We look at P30, we look at P60, we look at P90. And what is so interesting is that there is a dynamic change. Whatever you see at P30 is not the same as you see at P90. P30, I can tell you that the neurons, they know something is, P15, the neurons, they know something is wrong, okay? And they're so active, they're trying to repair it. They're trying to maintain homeostasis, and you can see all the genes upregulated. It's not because it is disease, it's because they're trying to overcome it. Let's say there is an ion channel, alpha subunit is mutated or it's leaky. They say, oh, I see the problem, let me fix it with this. So they're trying to fix it. But come P60, they say, I can't do this. And P60 is the time we see apoptotic pathways being activated. Come P90, it's clearance, it's clearance. So that's why it's very important for us to do the time course, because we are going to learn from the P15 cases. What is it that they are trying to maintain homeostasis, right? And what are also the proteins that change over time? So I'm going to come back to this, because the proteins that change over time is extremely important, because they give us an idea about a biomarker. And we were very lucky because some of those proteins were also detected in the blood. Haha. And they were also showing very high homology with human. Haha. So we said, all right, can we actually look some of the patients with upper motor neuron involvement and progressive upper motor neuron loss, and I collaborated with friends in Italy and Turkey. So we collected samples over time, 6 months, and we had like 5 or 6 patients. I mean, we didn't have many, but 5 out of 5 patients, okay, and these were all done blindly. We actually detected 6 proteins that show a trend going up, all in those 5 out of 5 patients. So now I'm trying to write grants, get monies to see if these proteins also hold true for a large cohort of patients, because if they do, that would actually be a very good biomarker that the drug companies are looking for. All right, so now I've talked to you about the RNA Seek and proteomics studies, and they begin to reveal the molecular basis. And the EM images that I showed you was structural damage, right? But then the genes that I showed you is actually the molecular basis of the destruction we observed with EM. And I think out of this, we will have biomarkers coming up, in addition to many other things. All right. So many people tell me, oh, but you're studying mouse neurons. I mean, this is not a mouse disease. And many people even say mouse may not be a good model, so you're wasting your time. Is that really so? So we actually had a paper in Nature Scientific Reports, and we find that whatever we see or observe in the upper motor neurons of the mouse models that we characterized, we actually see exactly the same thing in the bed cells of ALS patients. So if you don't compare mice to human, but if you compare upper motor neurons in mouse to upper motor neurons in patients, the translation is clear. Because at a cellular level, the cells are very, very similar, okay? Because they do the same thing. They again collect, integrate, transmit. The function is the same. The location is the same. The birth and maturation is the same. They are upper motor neurons in both species. So I think we have to focus our attention to the neurons, not really to the species. And again, here's one example to show you that this is what we have seen in different mouse models. This is in the wild type, this is in the SOD1, and this is in the UCHR1 I didn't have time to talk about. And here is in the other mouse models. And these are human samples. Look at this. These are apical dendrites in control cases, in sporadic ALS cases, and you can see all the vacuoles, you can see breaking apart, don't you? And this is exactly what we observe in the mouse. And when you have an apical dendrite like this, you're in trouble because other cortical neurons cannot talk to them. And I think this is one of the main reasons as well. Because imagine, your axons are dying, so you're losing the connection, right? Spinal motor neurons are losing the connections with the muscle, and your cortical cells, their apical dendrites are degenerating, so the cortex is not talking to them. I mean, the circuitry is broken. The London Bridge is falling down, right? So, but then how can we expedite drug discovery efforts if this is the problem? And I think we have to improve the translation. And then, as I told you, if at the species level, it may be different. Here's the human brain, here's the mouse brain, but at a cellular level, they're almost identical. And again, the translation at a cellular level, if you look at the apical dendrites in the mouse and in the human, it's the same. If you look at the mouse, that the cytoplasmic nucleus to cytoplasm transport is affected in the mouse, that's the same thing in the human. If you look at the mitochondria affected in the mouse, that's the same thing in the human. If you look at the ER having a problem, that's the same thing in the human. So I think by looking at the cell of interest is going to be very translational. So again, mice and human differ at species level, but open motor neurons in mice and humans are almost identical at a cellular level. So we think that translation at the level of cell and neurons will be translational. So that's why we were invited to write reviews about about drug discovery, and we had the idea that we can actually move the drug discovery efforts forward if we focus our attention to the neurons. And now we have the tool to do this, because I told you that the neurons become fluorescent, right? And so by crossing the reporter line that we have, we made these reporter lines. I told you before, many different lines, but this is very interesting. Because what I have shown you was in vivo, which means in the tissue. The wonderful, wonderful thing is when you actually dissociate the cortex, means like, pull every cell apart and put them in culture, the upper motor neurons still retain their GFP expression. This is wonderful for drug discovery, because in the dish, you will have cortical cells and neurons, and you will know exactly which one is the upper motor neuron. So when you add a drug of interest, a compound of interest, all you have to do is just observe. Are you happy? Are you not happy? Are you dying? Or are you doing better? So there is a clear readout if the drug is making the upper motor neurons happy or not. So that's the drug discovery platform that we are just developing. And again, I'm working with Dr. Silverman at Northwestern, and these neurons actually respond to treatment. This is also very nice. So of course, it's not only in vitro. You can give the drug to them in vivo, either oral or IP or gavage or something. Then you can also screen their cellular behavior or response. And again, I'm going to tell you a little bit about one of our experimental design with one of the compounds that we're working with Silverman. And you can, of course, do behavioral assays in these mice, right? And there's one assay that we think shows the upper motor neuron involvement, and it's the latency to fall when they're hanging upside down. So you take a mesh, you put them on the mesh, and then you lift it up, and they hang, right? And sometimes they want to go up. But if they don't really have that strength and have the ability to move themselves up, they will fall. But if the connection is there, they will hold, or sometimes they want to come back up. So this is a very good assay for us to see the integrity of the, or the health of the upper motor neurons. And we can see that they actually do better with our drug treatment. And now we also see that the upper motor neurons numbers are improved with a 100 milligram dose. And when you look at the misfolded protein in the cortex, these are the spinal motor neurons, here are the cortex, here it is. They co-localize, and this is the control, there's nothing. And now we also have another outcome measure to see if we can reduce them to this level. So it needs to be quantitative, it needs to be mathematical, it can't be just, oh, I think so, And then this, I think, is a quantitative measure. The other quantitative measure is the integrity and health of the apical dendrites, as I showed you before. And here, I can tell you that the apical dendrites degenerate, and with treatment, they actually revert back to control levels. So, coming back, I think looking at the integrity of the apical dendrite is another outcome measure, because we have looked at the percent of improvement, and so we are trying to develop these outcome measures which are reliable and that can be repeated and that can be used for treatment assays. So then our collaboration with Dr. Silverman, I think, has identified at least one compound that improves the health of upper motor neurons, and I think this is very exciting news for us. And so we are building onto this, and our hope is to actually move this forward. And we also have the drug discovery platform, and I'm working with multiple companies right now to investigate if their compounds also improve the health of the upper motor neurons. And we want this to be a requirement for preclinical studies to be initiated, because if this is a disease of the upper motor neuron, please study the health of the upper motor neuron, for God's sake, right? All right, so if I have time, I want to tell you a little bit about out-of-the-box approach that we have. So I talked to you about this. All right, so shifting gears, all right, changing topic. In neurodegenerative diseases, right, many genes are identified. So either you have the mutation, once you have the mutation of that gene, you develop the disease, right? So they are causative. Like you have the mutation, you have the disease. It's causative. And some genes are associated, which means you have the mutation, the chance of you getting that disease is increased. But it's not one-to-one. It's not direct. It's associated. And if you look, there are hundreds of genes now. For ALS, it was 148 since I checked. I think it's 150 now. For HSP, I thought it was 76. Now I hear it's 82. I think every day we find a new gene, and I don't think we identified all the genes for all diseases. Okay, how come different people with mutations in different genes develop very similar pathologies and very similar diseases, right? Everybody has a different mutation, and if you look at them, one gene is, I don't know, KIF5A, one gene is VPA, one gene is this one, and if you look, they are not relevant. They're irrelevant. How come different mutations in different patients lead to the same outcome? I mean, that's a big question, right? Okay. So we took an out-of-the-box approach. So we said every gene codes for a protein, correct? And every action in the cell is mediated by protein-protein interaction, right? Because proteins bind to each other, they are cofactors, they work together, blah, 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 and that's how things are done. And every protein has many different binding partners. Let's say you are a protein. How many friends do you have? Hundreds. You have different relationship with your friends, right? And you do your job based on your interactions with your friends, right? Imagine everybody in this room is a protein. And when I am here, I am the presenter. When I'm at home, I'm a mom. So I can be different people depending on where I am and what time of the day and where things are and everything, right? So one protein may also function differently based on where they are and whom they meet and who their partners are, correct? That makes it very complicated, but it makes it very interesting as well. So we said, if we know the gene mutations and if we know which proteins these genes code for, let's look at the protein-protein interactions. Let's see who talks to who. Let's see who is whose friend. So we actually looked 146. I want you to read this paper, please. We have 42 supplemental figures, 5 tables. One table is 46 pages with 1,400 references. It took me 5 years to write this paper. So please read it. Okay? Seriously. All right, so then we said, if we look at the protein-protein interactions, can we begin to tell who is whose friend, which game do they like to play, and when do they get upset? So I have an analogy to make. Let's say we all like to play marbles. We all love to play marbles, okay? But we have our preferred colors. Let's say I like blue, you like white, you like purple, you like, I don't know, black, you like red, whatever. So everybody has their preferred color, right? And in our pockets, we are allowed to take 5. So I take 3 blue, 1 white, 1 red, whatever, but I take mostly blue because that's my favorite. So everybody, fills their pocket with their favorite, correct? And then there's a call from the production facility. Oh, sorry, all marbles are fine, but the red one we're holding, we're not making red anymore. There's a problem somewhere. I don't care. I don't have red in my pocket. I keep my work. I still play. But whoever had red as their favorite are going to start crying because they can't play. That's selective vulnerability, because that makes you upset while others are still playing their games. Can I make it clear? So what we choose to play also begins to tell us what makes us vulnerable. So what canonical pathway you work on, or what is your goal in the cell, and if that goal you cannot achieve, you're selective vulnerable. Because let's say you're a GABAergic neuron, and your goal is to make GABA. You're an inhibitory neuron. There's a mutation in the GABA gene. As an excitatory neuron, do I care? No, I'm going to do it. But as a GABAergic neuron, you won't be able to produce GABA. You won't be able to be an interneuron. So you will be affected. But once you are affected and you can't inhibit me, then I will be affected later on. So it's the secondary effect. Do you see what I mean? So initial vulnerability is because what we choose to do, and we can't do them now, but then towards the end, the whole game, everything is going to suffer. Okay, so now, if I take you through this, all right, so this was covered in Science Trends as an interesting finding. So we looked at all the genes and their binding partners, one by one, imagine. Then we said, okay, there are, at the time when we did this, it was 147 genes. And then we said, okay, these are all the proteins, right? Let's say this is the mutated gene in the patient, and these are all the proteins that bind to the protein product of that gene. Okay, so these are his friends. This is him, this is his friends. This is this person, these are the friends. And guess what? Some friends are common. We call them the famous guys. Because there's one protein here which plays with this one. Oh, it also plays with this one. Oh, it also plays with this one. Oh, yeah. Oh, he's like he's a player. He's playing with everyone. He's everyone's player. So imagine that you have a mutation in that famous guy, then the interaction with other proteins will be affected, right? So we want to understand who is the famous guy, and what games does he want to play, and what games does he like to play. So rather than now mutations, genes, we're going into place. You understand, right? Okay. Oh, this is very exciting. All right, so then we say, all right, so these genes, who are you, where are you, what do you do? And who are the common guys? These are the common guys because they have, 17 binding partners. They are very famous. They almost bind with everyone. They're friends with everyone. So these are all the famous guys. And imagine these famous guys, when there is a mutation, there is a disease, which you would expect. Okay, so then first we did this. Then we said, okay, so these are how many proteins? 1,105. Then we said, proteins. What do you like to do? Which canonical pathway are you mostly interested in? In which canonical pathways do you take part in? And they start to talk. Oh, I really like to make sure lipid homeostasis is intact, and I'm working on this pathway, and the p-values are very high. This cannot really be luck, and this is the cell. This is the drawing. Look at this. All these proteins are in the protein list that we have. So it's basically taken everything. Oh, so that's important. And remember, I told you the Golgi and ER. ER is also where the lipids are made in the cell. And then we find, out of all these, who are again the common ones or the famous ones in this. Then they say, all right, protein homeostasis. We want to make sure that the protein homeostasis is in check. That's our goal. That's what we're working on. And then they say cytoskeletal dynamics. We want to make sure that all the actins, microtubules, and everything is in check. And I do this, I do that. And then there's a game. This is the game we play. This is the canonical pathway. I play a role in this. I talk to 5 people to do that. Then we say, do you see it? They begin to tell us what is important for them. Then we say, are there any upstream regulators? Let's say there's a boss. It says, do this, do that, or by changing the expression of one, can you change expression of others? Or one person modulates others. And there are, we find. Okay, so this is all bioinformatics, right? Then we go to human patients and say, these are the bed cells, and some of the proteins that come up, we say, because this is, imagine, this was all theoretical, all bioinformatics, all computers, right? But then we go back to human and say bed cells, bed cells. This is what the computer is telling me. Do you really have a problem with that protein? And the bed cell says, oh my God, it's so much, I accumulated in my cytoplasm, I can't get rid of it. And these were not identified before. So we identified at least 5 or 6 different proteins and different accumulations based on the bioinformatics that we have done. And again, we look at the human samples, this is control, look, bed cells are high, and now in the disease, they're almost gone. Some are upregulated, some are not present. So I have more examples to give, but then there's another one that you can see a lot of accumulation. So we think the problem may not be only in the mutation. The problem may be also at the protein level, and who talks to whom, and what games are altered. And we have to find out which games they like to play, and then based on the games they like to play, we will tell them, you don't like the red marble is not there, but is it okay if I give you the pink? Or is it okay if you tell them, for example, in your pocket you may have one red, or oh, he likes reds, can you please give your red to him? So we can start finding solutions when we understand what is the problem that they are actually upset about. So in my summary, I can tell you that upper motor neurons are not in the dark anymore. So we begin to understand why they degenerate, and of course more work needs to be done. And we are beginning to identify biomarkers for upper motor neurons which show the timing and extent of their degeneration over time. And we developed a novel drug discovery verification platform using upper motor neuron survival as a readout. And I think this is very important for drug discovery studies. And we are beginning to discover novel compounds. Sorry. Let's improve motor neuron health. So I may not have time to talk about the gene delivery approaches that we have, but we're also using AAV-mediated gene delivery very precisely to the upper motor neurons. And to be able to do that, we're engineering different promoters and different constructs so that we actually transfect not all neurons in the cortex, but mainly the upper motor neurons. And we actually published our paper in Nature Gene Therapy, and it's open to public. We paid $3,000 extra fee so that you guys actually can have access to them. Please look on PubMed. You type my last name, which is very hard, but if you type it, if you see our papers, you can download anything you want. So I would like to thank you for your attention, and I'm ready for your questions.